Yes, so today I'm going to talk about post-translational modifications. And proteins can be modified in a lot of different ways, and probably in more ways than we know. I mean, you all know about phosphorylation, which can happen at usually at serines, threonine, and tyrosines. Then you can have acetylation, methylation, ubiquitinylation. There is a lot of different, and these uh, modifications often modify the function of the protein or is used for some kind of signaling or change of function. So that's why we set out to try to analyze as many of these modifications as possible, either by focusing on a single protein and trying to characterize different modifications when it's in different states, or we look at it globally on the whole proteome. So the so the ones I mentioned so far are all uh, have something to do with the biological function. But then we also introduce modifications during sample preparation. And these are either reagents to quantify proteins or to make them uh, more amenable to mass spectrometry. For example, we usually alkylate cysteines because otherwise cysteines might react, are quite reactive and might react with other things or stick to the walls or disappear from the analysis. So that's why we usually alkylate them so that they become more stable. Then we also have a lot of modifications that are side products of sample preparation. So these are oxidation, deamidation, formylation, carbamylation. So these depend on the way we prepare our samples and the reagents that we use. Uh, so these are, and of course, we want to get to the biological modification, but we need to, when we analyze our sample, take all these other ones into account. So there is a very large space of different modifications that are possible. But the ones that people study and that are probably the most, uh, as far as we know, the most important one biologically, the list is not very long. And it's really, I mean, and phosphorylation is by far the most popular one to study. But then also acetylation, people have looked at ubiquitination and so on. But some modifications are easier to study than others, and that's also a reason why some are more studied than others. But this just shows a summary of some of the more common ones. And there is a whole set of modifications. So glycosylations are, I mean, could be very interesting, but actually hasn't been studied so far very extensively. There's been pretty much a few groups that have focused on it, and the reason is that it's a branch structure, and it's not as easy as just one mass change, but it could be a lot of different sugars that are linked to each other, and that makes it very difficult to analyze properly. But there has been some attempts at doing that. So this is an example of a phosphorylation. So this is similar to what Trixie talked to you about and it's probably, maybe it's exactly the same example that she used in one of the cases. So here we have the unmodified peptide, and we have calculated the B and the Y ions. And then in the case where we have a phosphorylation at serine 18, what we will have is that the B ions that are include this serine will be shifted by a mass of 80, and also the Y ions that contain the phosphorylated serine will be shifted. So the ones that are marked in red here are the ones that will have a shifted mass. And another example, if we have a phosphorylation at threonine 5, then again all the fragments that contain that threonine will be changed. So we an addition of 80 again, and those are the ones marked in red. So that's the way last time you were able to localize which residue was modified in the peptides by looking at the spectra. So then there is another issue with the modifications. When we don't know, when for example look at phosphorylation, and we have a peptide that contains 3 different possible phosphorylation sites, and then there are a lot of different possibilities. So each of them, in case we don't know anything from the beginning, each of them can be either phosphorylated or not phosphorylated. So this just shows that we have to test all these different possibilities when we do the search to localize, to identify the peptides and localize where the modification is. And then, so in this case, let's say that we test all these, and then we might see that this is the one that's phosphorylated. So another thing with a lot of these modification, including phosphorylation, is that if we do a global analysis of the proteome and look at all the peptides, there will be a very, very small fraction of phosphorylated peptides that we see, but this is usually less than 1%. So it's really what happens is that if we don't enrich for phosphopeptides, we don't see really many of them. And there are a few different ways to enrich. Most of them are based on some kind of affinity to either using IMAC or titanium dioxide. So this will enrich for phosphopeptide, and this just shows how you do it experimentally. You can do this in a tip. And so the idea is that here we can enrich for phosphopeptide, and then if we do that for the same sample as we just saw a few phosphopeptides when we looked at the global analysis, if we do this enrichment, we can see many phosphopeptides.
What percent does it go up to? Like, if it's 1%?
It varies, but it can easily be that more than half if you do it well. But it's still, but people are working very hard on optimizing several, all the different steps in this to really get a lot of. So another what one can do, and this can be also combined with phosphopeptide enrichment, is, for example, to do separation. So this technique called Hillick, hydrophilic interaction chromatography works very well and separates the unphosphorylated and phosphorylated peptide. So now the color here is wrong. So the yellow here is the unphosphorylated. And as you see that the early fractions contain mainly unphosphorylated, and the later ones have the first the singly phosphorylated and then multiply phosphorylated come even later. It's not a perfect separation, but it's definitely a clear trend that if you choose this later. So what people do is often they do both the separation pre-fractionation using Hillic and then all together with titanium dioxide. And then you can, at least in the later fractions, get mainly phosphopeptides. And then the same with the strong cation exchange. So there's many different ways to separate. You can also separate that the phosphopeptides in this case will be so the phosphosite is negatively charged, and then what you'll have the unphosphorylated peptides will be more basic, and the phosphopeptides will be slightly more neutral, so they will come out earlier. So there are many of these different methods that you can either combine or use one or the other to get more enrichment of phosphopeptides. So this is just one strategy that sorry, I missed the reference here. This is obviously from a paper, and I forget where it's from actually. Sorry about that. So this just showed that a SILAC analysis, and then here now they were interested in tyrosine phosphorylation, and against tyrosine phosphorylation there's actually an antibody that works reasonably well. So you can remove the phosphotyrosine, enrich for the phosphotyrosine antibodies and analyze those, or you can also split the sample and do analyze for the serine, threonine, and tyrosine together. And here they start with the cation exchange first into 12 fractions, and then do the IMAC enrichment for phosphopeptides, and then run 12 times 2-hour LCMS runs to analyze the phosphopeptides. So then one thing that often happens is that, so we mainly talked about collision-induced dissociation, where one lets peptides collide with the gas. And in that case, often what can happen is that one doesn't get very extensive fragmentation, but the main loss because of the excitation is that the phosphate group falls off. So in that case, we don't get very much information. But if we switch to another type of excitation, electron transfer dissociation, so in this case an electron is transferred and that gives the energy, we get a much richer fragmentation to be able to, and so what and the reason for this is that the phosphorylation is easier to break than the peptide backbone. So in collision-induced dissociation, we've also talked about that we get mainly fragmentation of the weakest bonds. while in electron transfer, the fragmentation happens much faster, so we get a much more even fragmentation along the backbone. So we talked a little bit about different methods to, experimental methods to enrich for phosphopeptides, and we can see that we can generate data where we can identify the phosphopeptides well. But then the other question is, how well can we localize? How well can we tell exactly which site is phosphorylated? This shows just a simulation of a case where we have a peptide with, in this case, 9 amino acids, and there are 2 possible sites that are phosphorylated. So now in this simulation, what we did was we randomly selected fragment ions from the peptide sequence, and then we select a different number of sequence fragments. So if we have few fragments, we never succeed with identification. And if we have many, we always succeed. So we go from, there's a transition from 0, where we never succeed, to 1. And at in this transition, what we have is that we sometimes succeed with identifying. So if we look at this, and of course we can then look at, for example, where we can identify 50% as a measure of how many, so looking at how well we are doing under different conditions. So this is now for identification. We have this 50% point at around 7 or 8 fragments. So now if we look at localization and first see if we have the distance between the 2 possible sites is 3 or larger, I mean in this case equal to 3, we see that that's far enough that we usually succeed with localizing. If we succeed to identify it, we also succeed to localize it. And for more than about, for about 47% of human tryptic peptides, this is the case, that we have the closest 2 sites for phosphorylation is more than 3 away. So quite often this happens. But sometimes, in about 1/3 of the cases, it's the distance is just 2 amino acids away. And then now what we see is we lose, it becomes more difficult to identify. And if we move to 1, it becomes even more difficult, that we need more fragments to successfully identify. And if we have a possible site, this is the most difficult case, we have 3, the real site is in the middle, and we have 3, 1 on each side that's a possible site, then in this case we need much more information to localize. So this is something to think about, is that if we have sites close to each other, we really need to have much better experimental data to be able to localize. So then the question is, can we start thinking about sort of a statistical framework for how to assign a confidence in assignments of phosphorylation sites? So we talked about how we can calculate different measures for how well we trust in identifications. So we're going to try to look at doing the same thing for modification sites. So in this case, it's we have an example peptide. We have 7 amino acids in each and 2 possible sites. So in this case, the modification can either be on the blue amino acid or on the red amino acid. Then we measure a fragment mass spectrum of this, and then we do the assignment. So here we see that the blue location, we have three peaks, these three blue peaks that correspond to it, and which correspond to so now the only thing that distinguishing these two sites are, of course, the fragments in between the two sites. So anything outside here on and on this side marked in gray, because they don't they help with identification, but they don't help with distinguishing the two. So in this case, we have three. We have a B ion here, and in the spectrum, and two Y ions. And for the supporting the red case, we have a Y ion here. So we see the intensities here. We have the support for that it's the red sequence is much less, of course, in this case. But we still should ask, what can we say that it's really only the blue, or is it a mixture, or can't we tell? That's sort of, those are the three possibilities here that we would consider. So then we can look at some other some real data here now. So here we have a peptide that it has two tyrosines in the middle, that and one of them is phosphorylated. So one way of looking at it is we can first take the first possibility that phosphorylation is on the first tyrosine, and look at the spectrum and assign the different peaks, and then move it, have the phosphorylation on the second one, and calculate those measures and try to assign peaks. And then what we see here is we have assignments to B3 and Y3 here, which is exactly the fragmentations in between here, but here we don't have those assignments. So in this case, we can quite certainly conclude that we have much more evidence that the phosphorylation is on the first tyrosine. So we can look at this in a different way, is that if we have assignment to the first, we match a total of 8 fragments, and in the case of the second tyrosine, we match 6 fragments, and there is a difference of 2. But we can also look at the intensity, and in this case we get the same answer, that we have more intensity supporting the first assignment than the second assignment. Now, so this is a very clear case, and we don't have really any specific evidence for that it's the second one. But if we look at this case, for example, so this is a longer peptide. It really has 3 main, so it can, from what we have most evidence for, it's either this tyrosine, this threonine, or this serine that's fragmented. And really there is another serine here, but that one has much less evidence for than the other ones. So if we look at these 3 now, so we have 15 that match if we put the phosphorylation on this tyrosine here. But then if we put it on this threonine, we only have 14, but there are 2 that are ions that are only that are not matching if we put phosphorylation here. So now we have contradictory evidence. Now we have to, and it gets even more interesting if we then look at intensity instead. Then the most intensity is supported by if the phosphorylation is on this threonine. And actually, in second place comes the serine, and only third place is the tyrosine, which was on top if we just looked at the number of matches. So now we have a contradictory evidence here, and it's not clear how to do this. And to give a little bit background, so most software that do localize, they use the number of matches and not the intensity. So in this case, they would probably assign it to the tyrosine, but not with probably not with a lot of confidence. So this is just to for you to think about that there is it's not localization is really not straightforward. So then one thing that one can do is to try to estimate the global false localization rate in a big sample. So now in this case we have a big data set, a reasonably large data set, and we want to know how often we localize in the wrong place. So this was the experiment here was we enriched for tyrosine phosphorylation. So we had most of our phosphopeptides should be tyrosine phosphorylated. But we also saw that we identified quite a few serine and threonine phosphopeptides. So the question is there a propensity for the enzyme to also enrich for those? But then what we tried was to do the localization of the phosphorylation for any amino acid. We said that okay, we're going to try to, even if we have a valine, we're going to allow it to, in our calculations, allow it to be phosphorylated. And that's what we see here. So these are all the amino acids and showing their amino acid frequency. And of course, most of these cannot be phosphorylated, like glycine or arginine and so on. So these we can calculate the false discovery. So we can say that this is the false discovery frequency that we see here. And not surprisingly, it's proportional to the amino acid frequency. Yeah.
Sorry, I'm kind of confused. What causes false?
That the data is not that in the MSMS spectra we have missing data and we have noise. So that's what that's what causes that we sometimes get it wrong. And by sort of letting in our assign any one amino acid at a time, but all of them, even though they can't be phosphorylated, we say that in our matching that, okay, now this time alanine can be phosphorylated. Then we can calculate get this relationship between amino acid frequency and false localization rate. So then, and we excluded tyrosine, of course, because most of them, because those are we know that those are most of the time correct. But by then going into this figure at the frequency of tyrosine in the data set that we have, which is much higher than the of course than the natural occurrence of tyrosine, we can read out and see that we have slightly more than 1% of our tyrosines that we've localized are incorrectly localized. Now then the question was, so the amino acid here is actually serine. So one thing that we wondered about is that we almost had 2% of our phosphosites localized to serine. But that doesn't seem to be an outlier. It seems to be pretty much in line with what in the general trend as a function of amino acid frequency. So probably most of these are just for localization because we have a high frequency of cysteine in our peptides. So now, so this was, we talked now about if we have a whole data set, but the question is, can we make a model and a way to decide how much we can trust an assignment? And so we're going back to our case with two different peptides. We have some evidence for each of them, but more evidence for one. So what we can do is we can make the assumption that 2 is the correct one. And then we now need to generate sort of realistic mass spectra in some way that are based on our data, but sort of selecting from the space that is realistic, and then estimate the probability that we get 1 by chance if 2 is correct. And then we can extend this if there are many, several candidates here that are high probability, then we can do the same. So what we want to generate is the sort of distribution of scores for assigning 1 as correct if in reality 2 is correct. And then we can from here just integrate the tail at the actual score that we get for 1, and then see what is the probability for that.
In the case of what exists in the sample, I mean, 40%, then what kind of?
Yes, so then we can take one step further. So now we're just asking what's the chance that 1 was false. But we can of course then go on. So now the next step is that if 1 is correct, what's the chance that we would also get that it would be a mixture of 1 and 2? And then we can turn it around and do the same kind of analysis, but now saying that we assume that 1 is correct. What's the chance of getting a score for 2 that's equal or larger to the score we get? So this is now the difficulty is of course to generate the spectra that are realistic, but sampling the random space, and that is still a work in progress, and we haven't really worked out how to do that properly. We have some ideas, but it's still not so this is but this would be the way if we could do a really realistic simulation of random spectra, then this could be a way to determine both, answer the question, is the first one correct and is it a mixture? So then, depending just on which probabilities we have, we get from this calculation, we can get either it's a mixture that it's 1 and 2, or that it's only 1, or we can't tell that it could be 1 and it could be 2. We don't know. So those are the possibilities that we get. So now, from so far we've mainly talked about bottom-up proteomics, and what people mean by that is that you digest with an enzyme and you look at the enzyme pieces, and you usually digest with trypsin, which gives you quite small fragments. But in particular, when you're interested in modifications, you might be interested in the way they correlate over a bigger part of the sequence. So, for example, if we do top-down measurement of this, so this is, let's say, a whole protein with 4 sites, then we could measure each of them, and there will be a lot of possibilities we have to try, but we could look at the correlation and see, for example, how often do we have this modified here and there. While when we do the digestion, we wouldn't see that. So this would be the bottom-up case where we digest, and we first of all, we don't see some parts of the proteome, and we also lose the correlation. So we don't know that maybe this protein shifts between 2 states. The one state that either nothing is, let's say, just this one is modified here, or this one. Or we can't, in the bottom-up case, we can't distinguish that from sometimes both of them are, and sometimes none of them are. So that's the, and also that often we don't see the whole protein. So just some general differences between bottom-up and top-down. So with bottom-up, we have small peptides, so they carry a few charges usually. Top-down, we have larger proteins, and they carry many charges. So it becomes one thing that we have is a dilution of the signal. So here the signal is, most of the signal is in 2 or 3 plus, but here it's distributed over many different charge states. Then also the isotope distribution is much wider, because here for a peptide we have a few isotope peaks. For top-down we have many isotopes, and we often can't easily see even the monoisotopic peak. And it's becomes a little bit difficult to say exactly what the monoisotopic mass is. So the other difference is that in bottom-up, of course, we have the small peptides that we fragment. In top-down, we have to fragment the whole protein, which is usually with collision-induced dissociation. We're more successful with small peptides, but when we go to larger proteins, people usually use electron capture dissociation or electron transfer dissociation. That works better in this case. So another thing that's very difficult to look at with bottom-up proteomics is alternative splicing. And most of the time we can only say, in global analysis, we can only say something about the gene. We can't really say which splice forms are present or quantify them. And the reason is that with bottom-up we get peptides over the different exons, but in this case we can't tell if this is one splice form or a mixture of several ones. But for example, in top-down, if we can see that exon 1 can be connected to exon 2 or exon 1 to exon 3, that there are two different splice forms. That would we wouldn't be able to tell in bottom-up if we weren't lucky enough to have the peptides that span the exon boundaries. So if we get these peptides, of course, then we can tell also. But the chance for that is not very high. So that's why people are trying to do more and more top-down. The problem, of course, there is that now you have to work with proteins, and they don't behave as well in chromatography as peptides usually, and also they're larger, so it's difficult to deal with them in the mass spectrometer. And proteins are very, some of them are very hydrophobic, and some of them, so it's not easy to not lose a large fraction of them. So, but even when you have these really poorly behaving protein in this context, if you digest it with an enzyme, there will always be a few of the of the peptides that come that are rather well behaving in mass spectrometry. So that's why bottom up is so generally used, because it's usually you get a few peptides, not always, but most of the time. But definitely there are questions that are would be much easier to address with top-down. And now there's been a few groups working on this for quite a while, and now there's more and more people working with top-down, and it seems to be that there has been more and more technology development, so it's possible to do it. Oh yeah, so we mentioned this already, so the correlation between modifications. So if we have two that are either always there or not there, so they're always together, either both of them there, that is a perfect thing that we can address with top-down, but with bottom-up we would lose these longer distance correlations. One one case has been histones, which are heavily modified, and often, and it's the modifications are used to regulate the nucleosomes. So, and here we really want to know also the correlation between, it's a typical example where we want to know the correlation between the modifications to be able to try to understand what how this regulation happens from the modifications. So for example, histone H3, its tail, N-terminal tail, is about, I think, 50 amino acids, and it's heavily modified in different ways. So this is H3, and just showing a few different modifications that can happen. So these are phosphorylations, methylation, acetylation, and this is the same for H4. It also can be heavily modified. Yeah, so what David Alice came up with was that there is a histone code. So these modifications of the histone tail serve as scaffolds and signals to different binding proteins to switch between states of transcriptional activation and inactivation. So that's why people have spent a lot of time trying to sort of look at how these modifications are in different nucleosomes.
So when we look at proteins, do we also enrich for modifications or something? I mean, when we do it, and we, let's say we are looking for the phosphorylation sites, are we going to enrich for phosphorylation proteins before we do the analysis?
No, so in this case, what we do, you can do it different ways. So when people look at histones, they usually enrich for the histone.
Okay.
So
Just raw protein.
yeah, so you focus on one protein and you analyze. So that's the other way. If you're interested in one protein to see all the different types of modifications, it's usually better to enrich for the protein. But sometimes people have done is to first enrich for the protein and then also enrich for phosphopeptides. So you can combine these in cases where you suspect, for example, that only 1% of a site is phosphorylated. I mean that the site is phosphorylated in only 1% of the protein. Then it's good to do both enrichment steps.
Because I just wondering whether the enrichments we have done for modifications specifically go for certain phosphorylations, let's say, other than the other ones.
Yes, no, so that's always a question, whether you mean whether all different phosphorylation sites will be enriched the same way, and it's probably not. I mean, the sequence around the phosphorylation site will definitely have an effect. We don't know what that effect is, but it will, yes. Yeah. So again, if we do bottom up, we lose the long-range correlations, which in this case for histones we do want very much to know what it is. So that's why it's better to look at, so in this case it's not the whole histone, but there is actually, people usually look at the cut after the first 50 amino acids and look at that. And this now, where you do some enzyme cleavage, but not with, use bigger pieces, it's called middle down. Not top down, because it's not the whole thing, but it's a big piece. And then you can see correlations, like here is illustrated. And you can, when you do this carefully, you see many different combinations. And people have gone in and tried to catalog these and see what, under different conditions, what how things these change. And so one way to cut, so there are some other enzymes, for example GLU-C. So this cuts on the C-terminal side of glutamic acid, and that's actually how one can get the N-terminal 50 amino acids of histone H3. So then what people, you get this distribution of charge states, and then you can go in and isolate the different pieces. So within this 10 plus charge state, one has all these different by different combinations of different modifications, so you get a lot of different modification states. So you have to go in and then isolate and fragment each of them separately. And then this is an example of a group of different modification states. So it's they all has 3 metal groups and 4 acetyl groups on the sequence, but in this case the metal groups, sometimes it's trimethylated here and sometimes 2 dimethylated and monomethylated over here, and then sometimes it's dimethylated here and mono. So all these forms you can then determine, and then there are other fractions where you get other pieces. So that was everything I had for today.